So here we see the 20 different amino acids shown a little bit different with ball and stick models. And what they're going to do is they're going to put those two of those amino acids together and create what's called a dipeptide. So when you take two amino acids like they're doing here and bond them together, you make a dipeptide. And they do that by taking off a hydrogen off one and a hydroxyl off the other, and they link them together and make what's called a peptide bond. And you're seeing the next one, so we've got a dipeptide, and now we're going to be forming a tripeptide. So there's three amino acids, and now a fourth one, so we get what's called a tetrapeptide, or now keep doing it, we get polypeptides. You can see water molecules are coming out as this happens. They call it the protein backbone. But the bond between two amino acids is called a peptide bond. And now they're showing as you keep forming these long proteins, water molecules come off every single time, and you grow these long chains of amino acids. They're showing them by single letters for the abbreviations to show that you get these really long structures, and that makes a protein. And the order of the amino acids is called the primary structure. So the fact that this one goes A, T, G, G, V, V, F, D, H, and so on and so forth means that that's the primary structure. The secondary structure we'll talk about later. So once again, a peptide bond is the amide bond between the carboxyl group of one amino acid and the amino group of the other, or the next amino acid. So we're putting together two different amino acids here. This one happens to be glycine because its R group is a hydrogen. This one is alanine because its R group is the methyl group. So we've got two different amino acids, glycine and alanine, and we're going to join those two together right here. We're going to take off two hydrogens off of this one, off of this ammonium group, we're calling it, and then this oxygen as well. And we're going to link this carbon to this nitrogen. And so that link between the carbon and the nitrogen, which is an amide linkage, they're calling it. It's an amide is actually between, or amide is between a carbonyl carbon and a nitrogen. Anyway, this particular right here, that is the peptide bond. So I might ask you to identify where the peptide bond is. It's between the carbonyl carbon and the nitrogen. It's that bond right there is the peptide bond. Okay, and now we have a dipeptide formed. Okay, because remember, amino acids are the building blocks for proteins, and we're going to start building a protein by joining the amino acids together like we saw in the animation. So again, two linked amino acids are called a dipeptide. We also have what's called the free amine side. So if we join again glycine and alanine, they're showing the same ones together. We get this, what we call the N-terminal. The N-terminal means it's the free nitrogen side. And then we get the free carboxyl side, or the C-terminal we call it, on this end. And the reason that that's important is because when we read this particular dipeptide, we would read the N-terminal first, and then we would read it in the direction of the C-terminal. So we always go from N to C. And so this one would be glycylalanine. The first one in the order is glycine, and the second one's alanine. And so the name of this dipeptide would be glycylalanine. Again, as we join these two together, glycine and alanine, here they're two single amino acids. When we join those two together, we get a dipeptide over here, and a water molecule is removed like we saw in the video as well, every time we create a peptide bond. And this is just another illustration. So again, we've got glycine here. Here's the alpha carbon, and one of the hydrogens. Here's the other R group hydrogen. And this is the oxygen that's going to come off. We're going to take off two hydrogens here and then join the nitrogen to the carbon. So there's our peptide bond, and there's our water molecule that came off. Okay, and again, we get glycine, then alanine, so it's glycylalanine, and then the order would be GA because we have the free amino side and the free carboxyl side. So we get the N terminus and the C terminus. Okay, we always read from N to C. And that brings us to this next idea of what's called the primary structure. Okay, the primary level, or the primary structure. The primary structure is just the sequence or the order of amino acids. And so when we get a polypeptide chain of 50 or more amino acids, okay, then we start to call it a protein. In this particular order, you can see like with these dashed lines, that is a peptide bond there. Here's another one. Here's another one. Okay, so we have 3 peptide bonds and we have 4 amino acids. We have this one right here, which happens to be alanine. And I'm starting with alanine because it's on the N-terminus side. So alanine is first. The next one is leucine. And then this one is cysteine with the sulfur here. That's cysteine. And then finally, methionine is the 4th one, and that's what this one is here. And I'm not so concerned that you know what these functional groups are and that you can, bring them down here like this. It's more so to see that there are 4 amino acids, okay? Alanine, leucine, cysteine, and methionine, and now we have what's called a tetra, because tetra is 4 peptide, okay? To get a full polypeptide of 50 or more and get an actual what we call protein, we'd have to be at 50 amino acids long, okay, in our chain. So here we're seeing a nonapeptide. Nonapeptide means that there's 9. These are 2 hormones that we find in the body. One of them is oxytocin. The other one is called ADH or vasopressin. And if you look at these 2, they're made by the posterior pituitary gland, or actually they're stored there. They're made by the hypothalamus. Anyway, these are ones that are found up in a gland up in your brain, the pituitary. And you can see both oxytocin and ADH have cysteine for the first amino acid. Tyrosine is the second, but then they're showing isoleucine for oxytocin and phenylalanine for, they're calling it vasopressin, and that's fine, but it's ADH. Okay, and then they share number 4, 5, 6, 7, and then 8, leucine for oxytocin, and arginine for vasopressin, and then 9, they share glycine. So 7 out of the 9 are the same, but at positions 3 and 8, they are different, and that creates 2 different hormones that have 2 very different actions. Oxytocin is involved with like childbirth. It's the hormone that causes the uterus to contract during childbirth, and then ADH is something that causes you to keep your water if you're dehydrated. It tells your kidneys to keep your water. It's the main thing that it does. Okay. But the point of this slide really is to show you that the primary structure is saying, okay, cysteine has to be first, tyrosine has to be second, and then for oxytocin, isoleucine has to be third, and so on and so forth. The order, the sequence of the amino acids is critical to its function. If it doesn't have that order, it won't do what it's supposed to do. Another thing we're seeing here is between amino acids 1 and 6, between these 2 cysteine amino acids, it's creating a disulfide bond. That's going to come up quite a bit. We're going to see that there's another linkage happening, and so it kind of gives it this funky shape, and that shape is critically important to the hormone's action. And so the primary structure, the order of amino acids, gives rise to its shape eventually, and that shape is really, really important for what it does. If it doesn't have that shape, for example, if we took out cysteine from number 6 and put in, say we just put in a proline here at number 6 as well, it wouldn't work anymore, and neither one of the hormones would work if we did that. Okay, so the hormones or the proteins in this case have to be specific in their order. Insulin was the first protein to have its primary structure determined. So the first one we ever sequenced the order and said, what are the amino acids and in order of this protein? The first one we ever determined was insulin. Insulin's a hormone that is made by the pancreas, and it regulates the blood glucose level. Insulin is made from 2 different polypeptide chains. So there's actually 2 different chains that make insulin. There's an A chain, which has 21 amino acids, and then a B chain that has 30 amino acids. So there's 51 amino acids in all in insulin. The 2 different chains, which are side by side, are held together by disulfide bonds, which we're going to talk more about disulfide bonds in the future. So if we take a look at insulin, here's the A chain. There's 20 amino acids. They have to be in that order. And then here's the B chain, and it has these 31 amino acids, and they need to be in that order. And then the 2 chains are held together again. There's a cysteine there, there's a cysteine there, and so these 2 chains, the A chain and the B chain, are actually linked together, and they need to be for insulin to work properly, and by these disulfide bonds here and here between these cysteines. And then the A chain, it's showing this disulfide bond here, and that's actually going to cause this green A chain to fold back, almost like a paper clip or a hairpin. It's going to cause it to bend backwards. And they're showing it in a straight chain, so it's easier to read, but really this is going to get a funky folded shape. All right. So human insulin has a primary structure that is very similar to cows and pigs. So, so cows and pigs are a little bit different at spot number 8, 9, and 10, and also at number 30. Okay, so in human insulin we have threonine, cows and pigs have alanine, and at number 8 we have threonine, pigs have threonine, but cows have alanine, for example. And we don't have to go through all the differences, but they're similar. So out of the 51, there's a lot of similarity, but there's maybe a little difference here or there between the two. Okay, so long story short, when we first found out about diabetes, and this is likely going to be a lab that we'll do, a video lab, I'm going to show you a video on the history of insulin, and then you'll answer questions about that because it's a really interesting story, actually. But once we figured out that insulin was causing diabetes, we started to use the pancreases of pigs and cows. So bovine insulin was used from 1922 when we discovered the cure until 1982. We take butchered animals and then extract the insulin from the pancreas and then use that in diabetic patients, and it worked pretty well for most of the patients. For like 90% of the patients it worked pretty well, but there was like that 10% that couldn't use it. They had an allergic reaction to it and it was bad. Today we use genetically engineered insulin, which is we give people human insulin, okay, and we make it in the lab. We don't take it from cows and pigs. It's way cheaper, it's way safer, and it's way more effective today to do it that way. We're going to talk about genetic engineering in chapter 21 with DNA. All right, so study check. Write the 3-letter abbreviations of the tripeptides that could be made from 2 glycines and 1 alanine. Go ahead and pause the video, and we'll check your answers in just 5 seconds. Okay, so what I came up with, so if I have 2 glycines and 1 alanine, I could start on my N-terminus side with ala-gly-gly, okay? Or I could go gly-gly-ala, or I could go gly-ala-gly. That's, I think, the ones that we can get. Maybe there's more, but those are the ones I came up with in just a minute or two. So, okay. So as we saw in the last video, we have this polypeptide chain with a primary structure of amino acids, and there's tons of amino acids in the chain, 50 or more. Sometimes there can be like 10,000, and so these are very, very long. What happens is, because of the amino acids in the primary structure, it creates what's called a secondary structure. And so there's 2 main shapes that proteins will take once we start putting the amino acids together. Either they're going to do this, and I'm showing this kind of in slow motion so I can talk and it doesn't go too fast, but this shape here you're seeing is called an alpha helix, okay? And it's this right-handed coil. It's kind of like a telephone coil, and now they're showing it with the amino acids in there with the R groups and whatnot. And now they took the R groups out and just made them these green little balls, so it's not so clumpy and bulky for you to look at. And they're showing you what stabilizes. These are called hydrogen bonds. They're going to show a hydrogen bond, I'm guessing, any second. Yep. Okay, so hydrogen bonds happen between the carbonyl carbon and the amine hydrogen, so the nitrogen hydrogen. There's an attractive force, kind of like a positive negative thing happening, and so these attractive forces are what caused this shape to happen. Another possible shape that you see is what's called a beta pleated sheet, and they're showing this little part here as an arrow to show you that it's going in this direction. But basically you get this kind of running in one direction and then kind of a twist or a turn and then going back the other way. Again, they're showing the side chains here, but again, the interaction of the sheet is held together with these hydrogen bonds, okay, between, again, the amino hydrogen and the carbonyl carbon. So once again, once we've determined the primary structure, which is the order or sequence, certain amino acids in a certain order will cause a secondary structure.
which indicates the spatial arrangement of the amino acids in the polypeptide chains. They're talking about the three most common. The two most common, again, are the alpha helix shape, which is the corkscrew shape we saw, which is like a coiled telephone cord. A beta pleated sheet is a zigzag pattern, which we saw, and then not as common, but we do see it in things like collagen. The triple helix is another structure, the secondary structure that we could see. Okay, so again, the alpha helix, like it says, is coiled and is held in place by hydrogen bonds between the amide groups, that's the nitrogen, hydrogen, and the carbonyl group of amino acids. So you can see here is the nitrogen, here's the hydrogen, and here's the carbonyl carbon, and that's what's drawing these things together is these hydrogen bonds. Okay, I don't want to make too much of like a deal about this, but that's what's causing the shape to happen, what's happening, why is it coiling like this? It's because of these hydrogen bonding interactions that are causing this this corkscrew shape to happen, this alpha helix. The same thing is true with a beta pleated sheet where we get amino acid chains going like this and then making a turn and then coming back the other way, and we're seeing these again, hydrogen bond interactions. It's just because different amino acids are on here that it gives this other possible secondary structure shape. Okay, and so we get this kind of back and forth weaving, if you will, making it into the protein, or the amino acid network, or the protein chain is then kind of weaved into this like sheet type of arrangement. Okay, again, this is found in things, fibrous things like silk. So silk would have this type of arrangement in silk protein. Then finally, triple helix. Again, it's not a real common secondary structure. It's actually it's made by taking three alpha helix chains and weaving them together. It's found in collagen, which is the most abundant protein in humans. It's found in our skin, and that's why they're showing wrinkles and things like this between these two people. Collagen is giving our skin some structural integrity. It's what makes our skin strong and so it doesn't rip. It's also found in bone, cartilage, and ligaments, tendons, things like that. And so here is kind of a illustration of a triple helix. All right, lastly, the learning check. Indicate the type of structure as either primary structure, is it an alpha helix, a beta pleated sheet, or a triple helix? Pause the video and we'll come back in a few seconds to go over it. Okay, so for A, polypeptide chains held side by side, that's the key there, by hydrogen bonds. I think that's talking about beta pleated sheets. So A is beta pleated sheets. The sequence, that's the sequence or the order of amino acids. That's the primary structure, okay? And so that's kind of what we're keen on is terms like that, the sequence or order. So is it glycine, alanine, glycine, or is it alanine, alanine, proline? What's going on? There could be all kinds of different arrangements. We have 20 different amino acids. There's lots of different ways we could order those. And remember, the primary structure is usually somewhere between 50 and 10,000 amino acids long. So these are long chains. The corkscrew shape with hydrogen bonds stabilizing them is the alpha helix, and then three peptide chains woven together like a rope, of course, would be the triple helix. All right, let me know if you have any questions, and we'll come back with tertiary structure and quaternary structure on the next video.